Our discussion is going to be open, so we are looking forward to receiving questions from participants. And you can either raise up your hand if you have a question, or just type your question here in the chat box, and I'll read it out for our participants. So we have four panelists, sorry, like I said. We have Dr. Giedrius Gasiunas, who is a Chief Scientific Officer. He leads research and development projects at Caszyme, and he is also a senior researcher at the Institute of Biotechnology of Life Sciences Center, Vilinius University, and a member of Young Academy of the Lithuanian Academy of Sciences. He obtained a PhD in biochemistry at Professor V. Zikny's laboratory, that's at the Institute of Biotechnology, Vilnius University, in 2012. During his PhD, he studied the mechanism of Cas9 protein activity, demonstrating that it is a programmable RNA-guided DNA in the nucleus. Dr. Giedrius is a co-author of 29 scientific publications, which are cited more than 3,400 times, and was involved in several Cas9 related patent applications. And we have Dr. Stephen Jones. He's from the United States, where he earned his doctorate in molecular biology, cellular biology, and biochemistry at Brown's University with Dr. Richard Bennett. He produced his postdoctoral research in CRISPR biochemistry and cellular aging with Dr. Ilya Finkelstein at the University of Texas. So there, he developed high throughput strategies for uncovering and benchmarking the targeting specificity of CRISPR endonuclease. In 2021, Dr. Stefan moved to Lithuania to become a founding group leader of the EMBL Partnership Institute for Genome Editing Technologies at Vilnius University's Life Sciences Center. He leads his group in his search to discover and characterize CRISPR endonuclease and other enzymes for genome editing. So this year, Dr. Stefan became Lithuania's first winner of the European Research Council's starting grant. So congratulations.
Thanks.
Yeah. So we have Dr. Heba Abouseada. She is a lecturer of molecular genetics at the botany department in the Faculty of Science at the Ains Shams University. She obtained a PhD in molecular genetics and biotechnology from Ains Shams University in 2014. Dr. Heba did her postdoctoral studies in CRISPR-Cas9 at Illinois State University in 2021. During her postdoc, she collaborated with the IPREFER, that's Integrated Pennycrest Research Enabling Farm and Energy Resilience Project, where CRISPR-Cas9 genome editing tool is employed to enhance biofuel production in pennycress and improve its tolerance to different abiotic stresses. And we have Dr. David Cisneros. He completed his PhD at TU Dresden in Germany, where he studied the folding and assembly of membrane protein complexes and the self-organization of collagen by atomic force microscopy. During his two postdocs at the Pasteur Institute in Paris and the Institute for Molecular Pathology in Vienna, he focused on the assembly and function of large macromolecular complexes, such as bacteria secretion systems and protein complexes, that regulates the folding and structure of chromatin in human cells. He's currently a research assistant at the Department of Molecular Biology at Umea University in Sweden, and he conducts research on protein secretion in intestinal bacteria. He's particularly interested in protein complexes that participate in the colonization process of human gut by the microbiota. So thank you so much for joining us today. And please feel free to drop any question you may have in the chat box or raise your hand if you would like to talk to our panelists. So let's start by asking would you mind telling us how you started with CRISPR Cas? How did you end up involving CRISPR Cas in your research?
Okay, so should I start first?
Okay, yeah.
According. Okay, so first of all, thank you for kind invitation to be here and also for kind introduction. Actually, I heard about first time about CRISPR in more than 15 years ago. In 2007, I was master's student at the time at Professor Vilinius laboratory in Vilnius University, and during my master's studies, I worked with all people in laboratory on another bacterial defense system called restriction enzyme. And actually, my supervisor, Professor Cisneros, read the seminal paper by Rudolf Barenguer and Philip Howard, where they show that actually CRISPR protects bacteria against phages. And it looked interesting to my supervisor, and he suggested just to select this topic for my PhD studies. So I would say it was quite risky path because for 3 years I had proteins purified, but they has no activity. But during that year, all the puzzles build it somehow, and we finally managed to elucidate the mechanism of Cas9 protein and show that it's RNA programmable DNA endonuclease. And then finally, after 5 years of my CRISPR path, I got my PhD. And now, as you see, I am my main reference is company. So after that, we patent the technology and licensed it to companies. During that, we started collaboration, a lot of collaboration with companies, and decided to separate those collaborations from this academic work, and that's how company called Caszyme started.
That's nice. Thank you so much for sharing that. Who would like to go next?
All right, you guys are making this awkward. I'll fix it. All right, I'll go next. Okay. So let's see. So the question was, how did we get into CRISPR, right?
Yeah.
So yeah, I guess I'll start when I first became aware of CRISPR, which was while I was in grad school back in the early 2010s. And I was afraid of CRISPR. I had no idea how it worked. And every time someone explained it to me, somehow it seemed more confusing rather than less. So maybe some other people here can share that feeling with me. But anyway, so that was grad school, and like you mentioned earlier, after I moved to postdoc at the University of Texas, I had originally gone there to study or to use high throughput technologies to study cellular aging. But as I'm sure many of you are aware, of course, sometimes you get started on multiple different projects because you don't know the progress of each different one, and maybe you just have a variety of interests. And that was really sort of my reason for getting into CRISPR, because I had a variety of interests. So I'm just showing you a little sort of like, I don't know what you call it, like an array of different topics that I had studied and have studied. And what you can see here is that CRISPR nucleases is only part of it. But what happened is there was this other project in the laboratory that people were trying to figure out how can we use a high throughput strategy to understand how proteins and DNAs interact with one another. And we had this sort of wild idea where we could use the flow cells that are used for next gen sequencing as sort of like a substrate for understanding these interactions. So basically we've got this flow cell with millions of different DNAs on the surface, and we could introduce fluorescent proteins and understand how those fluorescently labeled proteins were interacting with the different DNAs on the surface of this flow cell. And we thought, well, gee, what's a really interesting protein that interacts with DNA? I know, CRISPR, right? So we started with these sort of class 1 CRISPR nucleases, trying to CRISPR systems, and trying to understand how they were interacting with the DNA. And then, you know, we said, of course, you know, if you're a nuclease, you don't stop at just binding the DNA, you're actually going to cut the DNA. And so then we developed additional technologies that allowed us to understand how these CRISPR nucleases were finding the particular DNAs that they were interested in, and also cutting those DNAs. So basically, I had developed these high throughput strategies to understand these really critical features of the specificity of CRISPR nucleases. How do they tell what they want from all the things that they don't want? And I apologize, I have a bad habit of giving enzymes and molecules personalities. So you'll just have to deal with it, I guess, for today. But anyway, so this turned out worked really well, and I was very interested in this topic. And so when it came time to start my own group, I did just that. And I was particularly interested in this new center that was being developed at Vilnius University. So this is the Partnership Institute for Genome Editing Technologies, and it's a partnership between Vilnius University and EMBL. And so what they had done is recruited 6 group leaders from around the world to come here to Vilnius, Lithuania. And so basically we're working to discover, develop, and apply gene editing tools. And so I won't go into too much detail, but what you should recognize is that we have people here that sort of specialize in all these different parts of what completes the whole sort of gene editing picture. And so now that I've been here for about 1.5 years, I have a group that's steadily increasing in size and ability, and it's really exciting. And I'll just take this moment to sort of plug for a second, which is to say that we do have some open positions in the lab for both people that are basically at either graduate level or postgraduate level. And so I encourage you, especially if you have computational biology skills, to talk to me. So that was sort of like my career trajectory to where I'm at now. I will stop sharing because, of course, we need to make sure that everybody else has an opportunity to speak as well.
Yes,
so thanks for a moment.
Thank you. Thank you so much. Dr. Heba, would you like to go next? You're on mute. Okay, thank you.
First of all, I would like to thank you all for this kind invitation and for this organized event. I'd like to welcome the rest of the professors. It's very nice to be with you today. Concerning my experience with CRISPR Cas9, it started theoretically because I was in my master's and my PhD degrees. I used to work in different genetic engineering tools like gene gun or different bacterial transformations. But when I heard about CRISPR, it was about like 2012. I started to read a lot about CRISPR theoretically until I got my a chance to use CRISPR Cas9 in genome editing in Illinois State University. I had a postdoc fellowship from the Fulbright, so I traveled to the USA and I had my postdoc over there. It was about improvement of the biofuel production in Thalassemia arvens, which is pancreas, in Dr. Jones' Sadbrook lab in the School of Biological Sciences in Illinois State University. My work, if you'd like me to share like some slides.
Sure, if you have it.
Yeah, I do have them, but here. I worked on auxin responsive factors genes. These kind of genes, they were included in several signal transduction pathways in the development of different plants. And it was cited like after, if they got mutated, this will increase and enhance the drought resistance of different crops after mutating them, or down regulation of these genes. So I was lucky to be able of producing different mutant lines using 2 hitters for different genes for auxin responsive factors, ARF10, ARF16, and 17, and then double hitters for both genes 10 and 16, and 10 with 17 and 16 with 17, and a 4 hitters construct for silencing the 3 genes together. And I had, before I leave, I spent there 9 months. In these 9 months, I was successful, thank God, to produce 11 mutant lines. And these are some photos for Thalaspi advanced and the inflorescence for the new produced the mutants in line. I've left there, and one of the students should continue with this work to produce and to screen and do the drought resistance experiments in the field.
That's nice. Thank you. Thank you for sharing that. Okay. Yeah. Dr. David?
Thank you. Thank you very much, Joe. Thank you for inviting me. And yeah, so my story is less from the side of the developers of CRISPR, but really I started as a user of CRISPR. So it all started, I will share a couple of slides to explain you where it all began. Yeah, so I was studying at when I was working in the lab of Jan Michael Peter, we were studying a complex that is called the coesin complex, which participates in cell division. It's basically a protein that holds the chromosomes together, and then it's cut, and that allows that the chromosomes are separating during cell division. And we have found that this coesin complex had a role in folding and structuring the chromosomes during interphase, that is when cells are not dividing. And we have already quite a few really nice phenotypes that basically showed what we wanted, that this role during interphase, but we had no way of testing if we deleted the whole complex, whether we would see then the disappearance of those phenotypes. And that's where CRISPR came in, because the problem is that this complex is essential for the cells to divide. So one cannot create simply knockout cells. So what we did is that we used Cas9, and what we did is that we, using a repair template, we introduced a degron in the RAD21 gene, which is a subunit of this complex. So yeah, I'm glad that Dr. Abouseada introduced the auxin. So auxin is a plant hormone, and what we can do is that fuse a degron protein that responds to auxin, and that targets the protein to degradation. So what we did is that we, using CRISPR, we created a line in which the endogenous locus of RAD21 was tagged with a degron. So for example, here you have a movie of that. You see the cells, some of them that have the degron, after adding the auxin, the protein completely disappears. So the protein localizes to the nucleus. And yeah, so that's one of the projects in which I use CRISPR. And then when I moved to Umea, I came back to work with bacteria, which was also the topic in which I was working when I was working in Paris. And in that capacity, I started collaborating with Andrea Pujar's group, and she works also in bacterial secretion, and she works with a bacterium called Shigella, which is one of the deadliest intestinal disease bacteria, especially for children. And so it's one of those bacteria in which antimicrobial resistance is on the increase. So it was important to find new targets for antibiotics. So most of what is known of this bacterium has to do with a plasmid that codes for a bacterial secretion system. But Atin, a postdoc in Andreas' lab, was interested in studying genes on the chromosome. So I collaborated with Atin, and we established together a system to mutate Shigella in a medium to high throughput manner by using a Cas9 that has fused an AID domain. So AID domains are enzymes that catalyze, they're cytidine deaminases, so they catalyze the cytidine to uracil conversion, and then the repair mechanisms of the cells converted to T. So in that way, you can introduce stop codons at certain codons, and that are at a certain distance from the PAM sequence that binds the Cas9. And in that way, so what Atin did was to introduce base edited codons that coded for that were prematurely stopped during translation, and that essentially made loss of function mutants. And yeah, so we have published this already. It came out actually this month in M Systems. And yeah, so if you have any questions regarding cytidine deaminases or endogenous tagging of human cells, chromosomes, then yeah, you can ask me any questions. Thank you.
That's great. Thank you so much for sharing. We have like different areas of application of CRISPR Cas. That's so interesting. Please, if you have any question, you can raise your hand or type your question in the question and answer box or the chat box. So I'm just going to read that or let you speak. Okay, I think I see a raised hand. Sure, you can. Let me see if I can allow it to speak. Okay, yeah, you can ask your question, Martin.
Oh, so my question is very basic one, actually. And I don't know, any of you can answer it, I guess. So in my experience, I've seen that some guide RNA, they work better than other guide RNAs, but most of the studies on improving the CRISPR-Cas system, they just focus on improving the Cas9 enzyme instead. So what makes a guide RNA a good guide RNA, especially in bacterial systems? I know in some eukaryotic systems, there is a system of scoring. I don't know what that is based on, but especially in case of prokaryotes, it's very difficult to determine already or predict already that this guide RNA is going to be better than the other guide RNA. So every time I have to make a CRISPR-Cas mutation, I have to select different sites, choose different guide RNAs, and then hope one of them is going to work. So why do you think this is so far behind than improvement in Cas9?
Okay, I think Dr. Stefan raised his hand to answer that.
Yeah. I'm really happy to answer this question. I'm really glad that you offered this question as well. So I think, yeah, you've really highlighted sort of an important weakness that a lot of people have been dealing with in the current CRISPR technology. So what you should know is, of course, there are tools out there that are based on experimental data that are sort of computational in nature, and they're available on the web for helping you select your guide RNAs. Now, I say that realizing that as soon as you're working outside of a typical system, those tools become a lot less useful to you. And the reason for that is that a lot of those tools that are available are based on data sets that were either produced in human or mice or other, maybe another very common model that doesn't match what you're working with. So there's sort of two pieces to that, right? One is that if you are working one of these models, there's already verified guide RNAs that work for these different genes in the genome that you might be interested in. As soon as you're outside of that, those tools become less useful. But But there is work being done, including by my group, that what we're trying to do is find out what are those properties of guide RNAs that make them good no matter which organism you're in, right? So these would sort of be like the sort of foundational properties of guide RNAs that can make them better or worse. And I'll give you a really sort of basic example of this, which has been shown by a couple different groups, which is, of course, if you have some sort of, say, you pick a guide RNA and it turns out that it has either self-complementarity, or maybe it has some sort of complementarity with the rest of the guide RNA structure. Well, then what happens there is, of course, the guide RNA is going to make this sort of alternative structure that either doesn't allow it to interact appropriately with the CRISPR nuclease, or doesn't allow it to interact with its DNA target. So yeah, there are some things that are sort of basic that you can keep in mind, but that doesn't necessarily mean it's something that you want to have to check each time, right? So you avoid the obvious stuff, or maybe seemingly obvious stuff, but just realize that there's typically some sort of biochemistry behind this that is linked specifically to the enzyme, where we can improve in a way that goes across whichever organism or genome that you're working with. I think I mostly answered that, and I think David's probably got some good stuff to add. Is that right, David?
Yes, I think he raised his hand. Dr. David, you're on mute.
I thought I had. Yeah. So one important thing that Stephen mentioned that I reiterate is how these most of the databases that predict guide RNAs were probably not didn't use the organism that one needs. So it will be important to get the non-species specific data set. So that would be great when Stephen gets it. Now, I think on the other hand, at the same time, I think for the most part, even though they are not made in, for example, in E. coli or in the organism that you might be working, I think that the websites that predict gRNAs will give you they will select better guide RNAs that if you would randomly choose just any guide RNA sequence. So I think already there you are definitely much better using a website, and there are plenty, ChopChop and there's CCTop. I think those are the two that I use the most. And I think you'll be. I can put the addresses in the chat. I think you'll be better off using those than not using any, for sure. That I would say. And then regarding bacteria, so there is an advantage in working with bacteria because since their genomes are smaller, then the chances that you have off-target effects are smaller. And yeah, a lot of the websites that are predict guide RNAs, they focus a lot in off targets, and that's a very important feature for when you're trying to design guide RNAs, at least for human cells, in my experience. So I think you can get away with picking several guide RNAs, and I think many of them will be very good, and I think you have good chances of finding one very excellent guide RNA. And then the second thing that I wanted to say about guide RNAs is that redundancy is important. So you don't try to choose just one good guide RNA. I think you want to have several good guide RNAs, so you might as well pick as many as you can. Well, depending on the size of your screen, etc., as many as you can, as your screen can handle. Yeah, that's what I wanted to say. Yeah.
That's great. Thank you. Okay, Dr. Heba, would you like to say something about that?
Yeah, I think Dr. Stephen and Dr. David have covered almost most of the criteria that should be taken care of while choosing the guide RNA. What I need to emphasize that what happened with me personally, that we have to choose more than one guide RNAs, like 3 or 4 if we can achieve that. And experiments that would let you know which is the better. Sometimes it's like we choose all RNAs, guide RNAs for different genes on the same basis, but sometimes just one of them does not work as good as the others, so we have to repeat or choose from the rest guide RNAs we have chosen before. So this would make it easier for you. Never choose just one guide RNA. You should have plenty of them, as much as you can, or as much as they fulfill the characteristics for choosing good guide RNA. Yeah.
Thank you
Thank
so
you.
much.
You're
Thank
most
you for
welcome.
adding that.
Thank you.
So we have another question. What are the main challenges to CRISPR-Cas9 deployment, and how can we overcome them? Dr. Heba, I see your hand is still raised. Would you like to address this? You're on mute. Okay, I guess no.
I mean, I just forgot to lower my hand from the previous question.
It's
Yeah.
fine.
Thank you.
Yeah.
Okay, so maybe I can start with
Yeah,
the
thank you.
question. Since currently in company we are looking for new tools and trying to improve existing ones. So I would say that main maybe difficulties for employing, of course, is high activity in human cells or other cells, also of targets and delivery. Probably that's at least we are like trying to solve these issues.
Thank you for saying that. Does anybody want to add to what Dr. Giedrius said? Okay, Dr. Stefan.
Yes, so what I'd like to add is that one of the sort of outstanding issues is still delivery, right? So say we have the right tool, doesn't matter how great your tool is if it doesn't get to the place it needs to go. So the sort of some of the, depending on if you're in the sort of the research space or if you're in the therapeutic space, delivery might mean different things. So it's one thing to get the, say, CRISPR nuclease and its guide RNA into cells that are in cell culture. It's another issue when you have to get it to the right cells within an organ or a whole organism, right? So the challenge becomes even more immense when you're talking about sort of that whole organism level. And so one of the areas that's really being developed right now is the delivery strategies. So how do we do that? Sort of some of the traditional ways that maybe you could use in the lab is something like transduction, like these sorts of things. But as soon as you go into organisms, this is much more challenging. And so then we start thinking about delivery mechanisms like viruses that can be used, such as adeno-associated viruses, or maybe sometimes you can use lentivirus. But now we're getting into the place where people are starting to use nanoparticles for delivery. They've been engineering sort of what was the terminology that they use? Basically, a sort of a hybrid between both a packaging that's based on viral particles and then also the actual thing that you're trying to deliver. So it's a hybrid of the packaging material and the delivery material all in one, and that can be delivered as well. So that being said, I think even with these advances that we have right now, this is still one of the biggest issues to implementation. And it's the reason why a lot of the therapeutic applications that we see right now are targeted either towards organs that are very easy to deliver to or able to physically access. You know, when you think of, say, one of the recent CRISPR technologies, which is fixing a type of blindness, well, your eye's right here. You have no problem getting to it, right? Now, if you're talking about some small cell population in some small part of your body, suddenly you can't just, you know, stick a needle in and hope that you're good, right? Now you have to figure out, how do I get these guys to go to those cells and those cells only? So that's what I see as one of the major sort of limitations that we're still working on, and is well worth spending some time developing the technology.
Okay, thank you for thank you for sharing. If nobody wants to add to that question, I think we have another question. So it's, what are some of the prospects of CRISPR in the environmental field, especially with sustainable development goals being on the agenda of the world today? How can CRISPR serve the goal of ending poverty and food safety, or even climate change? Okay, Dr. Heba.
Yeah, because I mean, concerning the sustainable development goals for food safety and for the environmental issues, I think for food safety, one of the main perspectives should be increasing the tolerance for different crops, increasing their tolerance for different abiotic stresses, especially drought, because drought will save both the crops and enhance their productivity, along with saving a lot of used water for watering crops. So it will double hit two advantages together, as saving or increasing the crops productivity, along with saving water for the human use. Concerning another issue for the environment, also the enhancement of the biofuel production and the decrease of carbon dioxide emission. So this will also be implemented very well to serve the SDG goals, or the Sustainable Development Goals, concerning saving the environment from the carbon dioxide emission and the greenhouse effect.
Thank you for sharing.
You're
Dr.
welcome.
Giedrius, would you like to add something to that?
Yes, I would like to add from as a perspective, because I a little bit involved in this discussions how all these modified organisms and plants should be regulated in European Union. Maybe I can comment a little bit on that, and I think everyone agrees that theoretically, like CRISPR can be used for development of new plant new plant species for agriculture, and most probably it's needed a lot, because you know that climate change, also number of human constantly increasing. It means that like and this nature most probably the most suffers for that, because we need more and more land for agriculture to maintain the same like food production. And therefore everyone, I think, agrees that something new should be done. But problem is, for example, in EU now that actually all all edited plant species are considered like GMO. You can delete only one nucleotide, but it will be regulated as genetic modified organism, and therefore it's like, I think no such products are yet in European Union. So from and seeing how discussions are going in this European Commission, I am not very optimistic that something will change soon.
Okay, thank you for sharing. I think I saw somewhere that they were talking about using excluding in vitro plants gene editing techniques from GMO rules according to the European Union laws. I don't know how applicable that is currently.
So it's it was like separate countries could exclude it, whereas could exclude it until 2018, I think. But then there was like after that there was like some explanation from European Court that actually if something was done on purpose, it should be according to laws, it should be regulated as genetically modified organism. And now everything in limbo, so in discussion level. And
wow.
for example, in US now and in other countries, if modifications are identical to natural ones, it's not considered as GMO.
Thanks for sharing. I think Dr. David wants to add something.
I think Atin also raised his hand first.
Okay,
it's all right. Go ahead.
Okay. Well, no, I just wanted to say that yeah. So there are two aspects to this question. So I think on the one hand, what has been said is very important. So if we could generate better crops, and yeah, I think there would be a lot of benefits. And if it would be legislated in a useful way, it would also be useful. But I think also one important advancement that CRISPR brings is by basically studying what to modify, right? And I think that is something that we should all keep in mind, that at least what I consider one of the biggest contributions of CRISPR is the ability of many of us being able to, like for example, in my case, modify a gene that I could have not modified, right? And at the same time, or modify thousands of genes in one go, and finding out that phenotype that you might want to control. So at the moment, maybe in Europe it's not possible to modify them, but then you could find a product that modulates it. Or I think this side aspect should not be forgotten in trying also to combat climate change and improve our ability to fulfill the sustainable development goals. Yeah, that's what I wanted to say.
Thank you so much. Aten, you can.
Yeah, I just wanted to add something to the discussion.
Okay.
So this is not the first time that I'm listening from somebody who's involved with CRISPR Cas complaining that European Union has such strict rules that even if you delete one nucleotide base and it is characterized as GMO and not allowed to have, you're not allowed to produce it commercially. But what would it take for policymakers to change their mind? I mean, it has to come from scientists, right? What can we as researchers, as scientists can do to, I don't know, change their mind, introduce them to the concept that it is not that, or are we sure that it is safe enough? How can we as scientists make sure that the policymakers listen to us and have an open mind for this?
That's one good question. I think Dr. Stefan has his hands raised. Maybe he can go for that question.
Yeah, I'll do two parts because I want to add one thing to the last part,
Okay.
but then I'll answer this first. So, I think this is something, so, like Mary mentioned earlier, I'm from the United States, right? And so one of the things that I learned through the pandemic was realizing part of our role in society as scientists is also to be advocates for scientific advances, because if we don't say something, someone else will. And that may not be the message that is actually true or should be delivered. So it's a responsibility that we share in our profession, and we need to recognize how we can become more effective communicators so we can make sure that the right messages are being shared. And also it's about realizing who do those need to be shared to. So, one thing that I was really excited about, I was recently at a biotechnology conference, particularly, focused on food crops recently. And at least within that crowd, there was an understanding among the different stakeholders there is man, we really kind of screwed up here because we didn't say the things when the things needed to be said. And so now we're sort of playing catch up, right? But this is a super critical game of catch up to play. And it's one of these things that we can't just say, well, we screwed it up, so now I guess we'll just live the rest of our lives with it screwed up like that. We do really have to do the hard work. And I say we because of course it's the scientific community, but also it's communicating with people that aren't scientists so they can help sort of share that message. I don't know if that sounds a little too like kumbaya or something like this, but anyway. And the other thing I want to mention about the prior question about environmental and sort of gene editing being equitable, food poverty, food safety, etc., is that I want to recognize that CRISPR technology actually has gone a huge distance in making gene editing accessible and more available, because it wasn't so long ago, remember, it was only maybe 10 years ago, that the technologies that could do the same thing that CRISPR does right now, they were thousands of times more expensive. It was something where even a standard lab couldn't make the gene edits that they needed to make, where something like making a model line for studying whatever topic was something that was going to take months. Now it can take days or maybe weeks, right? So the landscape has changed significantly in a way that does make gene editing more accessible. That being said, the other half of this is, of course, is a very lucrative business, right? And so we do have to recognize, of course, we need the involvement of companies like Caszyme in making sure that these products are available to people that want to use them, but also thinking about making sure that the prices are sensitive to the places where people are and how this is sort of like really a tool for mankind and not just for the most wealthy nations. So I'll add that on.
Thank you for sharing. Dr. Giedrius, would you like to add something?
Yes, I want to say that I agree with Stefan that also the communication by scientists and maybe learning is the most important thing what we can do, because I think in current times everyone lives in some kind of bubbles and it's not so easy to overcome that. So, and especially I hear that for example with EU regulations that actually European Commission tried to listen of like voices of all stakeholders. And for example, some farmers say that I want to do like my grandparents did, but they do not understand that they live like, because there now stuff is more efficient. You by doing that you live like bigger and environmental footprint. Yeah.
Yeah, that's so true. Thank you so much. We have another question. How can I have efficient transduction of primary human T cells? Any advice on what would help me? Who would like to address the question?
Well, maybe I can say something. So primary cells are one of the toughest challenges, I think, for CRISPR, and I think developing new ways to deliver Cas9, I think is going to be super important. So it's good that companies are working on this. I think for the moment, the published strategies are dealing with delivering. For most cells, viruses are not an option. So retroviruses or lentiviruses are not an option. So they have mainly as an option electroporation. So then you can electroporate DNA. But there has also been good examples of electroporation of directly protein, so purified Cas9, already pre-assembled with guide RNAs. And finally, there are also some examples of primary cells being electroporated, but with mRNA, so that the protein can be produced from the messenger RNA. But yeah, I think in general, I think primary cells are the biggest challenge. So in that case, if you decide to use electroporation of protein, then you need to have a way to produce that protein yourself at this moment, and that is requires a very different infrastructure. So yeah, I think that is going to be very important when there are commercially available kits to deliver cells with the either protein, messenger RNA, or DNA in more efficient ways. Yeah.
Thank you for sharing. We have another question from Carolise. So often commercial Cas9 is sold with stabilizing additives such as glycerol. This is sometimes annoying because injection needle is easily clogged. Do you have special variants of Cas-like enzymes that would remain highly active even in the absence of these sticky stabilizers? Okay, Dr. Stefan.
Great. So I think what you're actually coming up against here is difference in goals, right? So actually many of these enzymes, including Cas9, do just fine in the absence of something like glycerol. The question is, can the company still be able to promise you that this enzyme will stay stable for this amount of time and sort of be able to deliver on the other expectations that maybe you have as a user? In this case, of course, something that's easy for me to say is, oh, just go ahead and make it yourself. I realize that's not always a possibility for everyone. But what you can consider is either maybe talking with a company that can provide this, or maybe some other type of CRISPR nuclease, maybe has different goals in mind. So for them, it's not such a big deal if they have a particular user and you know, you say, I know this is not going to last as long because of this, right? So you may be able to talk to some of these companies. I've never tried it, but I do suggest it. The other thing is you might try delivering something besides the protein, right? So this sort of connects with what we were talking about in the past, but you might be able to say deliver an mRNA that encodes your CRISPR nuclease, and maybe that's sufficient, right? So then you don't have to worry about the stabilizer being there, but you can still have a little more control about what's being delivered to the cell.
Thank you. Dr. Giedrius, would you like to add something to that?
Yes, so I think a reason why most of reagents companies use buffers with glycerol is because then enzymes could be stored in -20 degrees, so in simple fridge, which is convenient both for shipping and also storing it for keeping it for a long time. So, but from my experience, I don't know if some companies provide that, but you can like froze protein in -80 in any buffer, and usually it remains active. But then you need to it do not survive like freezing throwing cycle, so it could be thrown only one time.
Thank you so much. I think Miriam has her hand raised. Would you think you have the access to speak now? Miriam?
Hello?
Yeah, we can hear you.
Yeah, I just got super excited when we were discussing science communication and how important it is for scientists to like advocate for science and like talk with the public about it. And I wanted to say how important this webinar is and how being together as scientists and talk about these things is so important. So I wanted to thank each and everyone who like invested in his time and like effort and is with us today to communicate science. So yeah, thank you.
Thank you, Miriam. Okay, great. Just a quick reminder, you can raise your hands and would allow you to speak, or you can just drop your question. So there's another question to Dr. David. So how long were the homology arms that you have used for the auxin-inducible Degron system?
Yeah.
Do you have any recommendations about this? In the literature, it is quite variable size. Does it depend on the size you want to introduce on the genome?
Yeah. Yeah, actually, that also reminded me that I had something to say about the previous thing, but I think this is also fitting here. So, well, I used 1,000 base pairs homology arms. 500 worked also well. But the thing that I wanted to say is, I think Stephen touched on it, that it also depends what you need, how many cells do you need. I think that it also depends. Yeah, your delivery system might be very bad, but if you have a powerful way to select your cells, then you can get away with it. So that's, I think, one important thing to think about, which was the case for what I did with the auxin Degron. So we had, well, it was also the beginning. So it was some time ago, and it was really the beginning of CRISPR. So I'm sure that now there are much better systems, but we could only get edited cells with an efficiency of 0.1%, and when we were lucky, 1% of the cells. But because we were also using a GFP tagged version, so we could very easily select the cells by FAC sorting. So, and because anyway we had to grow them from single cells, we basically just needed around 200 cells, right? 296 well plates would do the trick. So yeah, so it yeah, delivery systems also might not matter too much if you have a good way to select. And now there are also, if you go to a gene, there are vectors that that have tagged versions of Cas9. There are vectors that have antibiotic resistance cassettes. So yeah, you might be able to get away with low delivery efficiencies. Yeah.
Okay, great. Thank you. I think Dr. Stefan wants to add to that.
Yeah, I just want to reiterate what David's saying. You know, you've got two different strategies. Either you try to get a lot of what you want, or you try to kill everything you don't, right? So recognize when you can do that strategy and use it to your advantage. I actually wanted to ask a question if I can. Is that all right?
Yeah, sure.
So basically, you know, I saw the name of our thing today, and it's Advances in CRISPR, CRISPR biotechnology. I forgot exactly what it was, but anyway, I was just wondering, maybe we could go around and each of us could just maybe share a new advance in the field that they've heard of that they think might be interesting to the audience. What do you guys think? Yeah.
That sounds good.
I think you have one in mind that you want to share.
No, I'll wait. I want to see what you all share first.
Then you could start.
Okay.
Okay. All right, fine. So, you know, one of the things I was just reflecting on as we've been going through this panel is I think we've been talking a lot specifically about using CRISPR nucleases for gene editing, right? So basically we're trying to use something that's going to cut DNA, and then somehow we're going to depend on cells to try and repair those cells of the genome in some way that's favorable for our outcome. So I think, you know, some older technologies that we should be aware of is that we don't always have to cut our DNA. We can use technologies like base editors or prime editors or transposon-based systems, all these different things that are going to maybe allow you to perform a gene edit while avoiding the toxicity that can be associated with the double-strand break. The other thing is we can consider tools that instead work on RNAs or perhaps the expression of a gene. So we can think of either interference systems or activation type systems where we're going to regulate how the gene works. And in those cases, we're only dependent on the CRISPR in order to get to the right gene, but not necessarily to cut that gene, rather just to regulate it either through maybe blocking a transcription factor or maybe modulating some sort of chromatin decoration that might be there, something like that. Or we can think of stuff that maybe is like some of these enzymes that particularly target RNAs, right? So we can imagine if we just need to knock down expression of a particular product, then we could destroy the RNA. So there's some of these CRISPR systems that instead of targeting DNA, they will target RNA, and those are becoming very useful tools as well. So don't think of CRISPR as just Cas9 and as just a tool only for cutting DNA.
That's very insightful. Dr. David, do you want to add something new?
Well, I actually maybe I should shoot myself in the leg at this point and say that there are also, I mean, but for some technologies, you don't have to use CRISPR, right? So that is another. I have talked to people that seem to want to use CRISPR just because and not because they really need it. So that is maybe one warning that maybe we should also do. So if there is a method that works really well for the one thing you want to do, then yeah. But if you want to increase throughput, then certainly CRISPR is one thing that will be able to help you with that. Yeah. Maybe what I wanted to talk more about is because you mentioned them all, Stephen. I think you said everything. You said you mentioned RNA targeting.
Maybe some delivery methods or some new applications or some new therapeutics. I don't know.
Yeah.
Diagnostic strategies.
Think about it. Yeah, well, that, yeah, You're mentioning them all. Yeah, I wanted to talk more about trade-offs. So I think in particular about base editing, which is what we've been using lately. So one thing that I think one needs to consider is, so again, on targets and off target effects. But when you're doing base editing, you also need to consider in a specific base editing. So the base editors can also go awry and start, yeah, changing genes.
Can you remind
where
us
you
how
don't
they
want
work?
to.
Can you remind us what a base editor is and how it works? Just
Yeah, I have a great slide for that. Yeah, I think you can see it now. Yeah, so base editors, well, for example, here we have an example of one base editor, which is AID. So AID is an enzyme that in B cells participates in the hypersomatic mutation in humans, and that helps the maturation of the antibodies so that they have more affinities. But they are basically belong to a family of proteins that most of them introduce deamination in cytidine, and as I said, that can catalyze in the DNA a mutation. So that is what it has been exploited by fusing these enzymes to Cas9. Then if Cas9 is, for example, a catalytically inactive Cas9, then it can target the Cas9 to a particular locus of interest, and then you let the base editor to introduce a mutation, hopefully very well designed so that it could introduce, for example, in the case of what we did, a stop codon, and in that way generate a loss of function mutant. But there are several things that one has to consider when using base editors. So for example, Cas9 in the absence of guide RNA has a certain affinity for DNA. So it likes to bind to DNA more than when it's bound to a guide RNA. So in that case, if you have a control where you want to measure the rogue activity of the ID, I think it's better to use a guide RNA that has no target rather than no guide RNA, because then you might have a false positive readout there. And then, yeah, so, but in the absence of, yeah, in any case, these base editors might bind to DNA without Cas9. They will have some affinity for DNA. So the one that we are using, for example, which comes from lamprey, primarily binds to single-stranded DNA. So at least it needs an open complex. So we're thinking that that reduces the levels of in a specific base editing. But we have observed situations in which there is, well, yeah, more mutagenesis. So yeah, that also, I think it is good to remember that for any base editor to fix the mutation, it depends on the repair mechanisms again of the cell. So going back to what Stephen was saying before, so you depend very much on the repair mechanisms of the cells, because if there's a C2U mutation at the DNA level, well, the repair mechanisms can send it back to C. Only when it fails, it will fix the C2T transition. So that will be also species specific, and that might change from organism to organism. And therefore, the levels of, let's say, in a specific or off target will have to be determined for each base editor and for each organism, like which conditions, how much you express the Cas9, how long you leave the base editor in the cell, etc. Yeah, that's what I want to say.
And that is great. Thank you for sharing. Dr. Giedrius, would you like to drop something new?
Yes, so maybe I would like to note and say that I am surprised in a good way about advances in therapies, especially base editing, which you know is like synthetic tool. And recently, I think I read this in December last year, there was a story about girl which had incurable lymphoma. Actually, base editing was used to modify cells to make donor cells like, resistant to therapy, and this helped to use CAR-T therapy to this girl. So I think for me, it's most amazing thing that it's working and it's already like yes, good successful example.
Okay, that's great. Dr. Heba, would you like to tell us something new?
I think everything new has been said. Start with Dr. Stephen, followed by Dr. David. I think I don't have any more to add. Thank you.
Okay, that's great. Thank you so much. I think we're a little bit past our time. So I would just like to thank you all for attending, our panelists for agreeing to speak during this discussion, and also all the participants for tuning in from everywhere in the world. Thank you so much. It was really nice having you guys. And then we say a very big thank you and bye. Bye.
Thanks, everybody.